That's really one of the kindest introductions I've ever gotten, so thank you for that. Just so people know, I do have my video turned off at the moment just because my internet connection sort of gets a little unstable if I have it on, so I apologize that you can't see me right now, but hopefully everyone can hear me okay. So as Darren mentioned, I'm going to be presenting the results of HPTN 083, which is a PrEP study that was done, as Darren said, in cisgender men and transgender women who have sex with men. And the idea was to see if long-acting injectable PrEP could compare to daily oral Truvada for PrEP in those study populations. So these are the co-authors on this abstract. It's a lot of names, but it really took a village to do the study, and this isn't even a complete list. I'll show you a complete list at the end, and you can see how many people were involved in this study. It's really super exciting. These are my disclosures, and this is what we're going to talk about today. I'm just going to review a little bit about PrEP background and why we thought the study was an important study to do. And then I'm going to tell you a little bit about the study design. Then I'm going to show you the results. I'm going to show you who enrolled in the study, and then what happened in terms of its ability to prevent new HIV infections. I'm going to tell you a little bit about the safety of the medications. I'm going to tell you a little bit about the people who got HIV despite being in this trial and what we know about them, and just some other interesting things that I think are important for us to follow up on, and then what we're doing next. So I hope that makes sense. And I'm going to do all that in about 20 minutes. So get ready, buckle your seatbelt. Okay, so the reason the study was important, what these pictures are is these are the estimates of how well PrEP works in different populations in the previous studies before HPTN 083. And each figure tells you the sex at birth of the people in the study, and how filled up the figure is in blue tells you how protective the Truvada medication was against new HIV infection compared to a placebo pill. So you can see that the numbers are kind of all over the map. And of course, what we've learned from all of these studies is that we know that PrEP works really well if you take it, and works less well if you don't take it as prescribed. And so a lot of these very different results in different populations were most driven by how well people took the medication. And some of you are probably wondering, what about the little guy in the bottom right corner wearing the French beret who looks like a mime? And the reason he's stripy like that is because that was what's called on-demand or 2-1-1 PrEP. It wasn't daily dosing. It was people taking PrEP just before and after their sexual events. So that is known to work as well, even though it hasn't made it into most guidelines or regulatory approvals in the US yet. There are also some newer PrEP agents, including the recent European Medicines Association approval of a dapivirine ring for HIV prevention for cisgender women, which is tremendously exciting for additional options for women, and the Discover study that showed that TAF FTC or Descovy can also be an option for PrEP for all routes of exposure other than vaginal exposure. So PrEP has really moved on past just Truvada. We now have new ways to take it. You can take it on demand. We have a vaginal ring that has approval, and we have an additional agent in some populations for oral daily dosing. What 083 did was to say, we know that there are some populations that either don't like or have trouble taking a pill. I know there's a lot of words on this slide, so I beg your forgiveness. But the idea here was we're taking a new medication called Cabotegravir that's available as a long-acting shot. It's also available as a short-acting pill. And we're going to compare it to the standard of care, which is daily oral Truvada, again, in cisgender men and transgender women. And the study design was what's called randomized, double-blind, double-dummy. And what that means is everybody got both the Cabotegravir and the Truvada throughout the whole study. One was real, one was a placebo or a fake, and neither the participants nor the people conducting the study knew who was receiving what until the study ended and was unblinded. We did this at 43 sites all over the world. I'll show you in a second where those sites were. We enrolled cisgender men and transgender women age 18 and above who were at elevated risk for HIV infection. They were generally pretty healthy. They did not have hepatitis B or C. And there was no reason that they couldn't have injections in their butt muscles, because that's where the injectable product has to be administered, and they couldn't have a history of seizures also. The study was originally planned to enroll 5,000 people. I say originally because I'll tell you more in just a second what happened. And pre-specified in the study, we planned that at least half of the study population would be under the age of 30, at least 10% of the population would be transgender women, and at least 50% of the participants who enrolled in the US would self-identify as Black or African American. And a lot of people have asked why that we had those pre-specified metrics for the enrollment. And the answer from our standpoint was that we thought that historically these populations were underrepresented in PrEP trials, but they are disproportionately affected by HIV. And so finding PrEP and prevention medications that were acceptable, safe, and effective for our most at-risk populations, our most vulnerable populations, was critical to any prevention intervention actually being useful. So that was the reason we set those metrics before we started, and we had tremendous partnership and help from a wonderful team of community program managers and community educators, both at the HPTN who conducted the study and at each of our sites, as well as the HPTN Black Caucus. And we'll hear a little bit more about that group as soon as I finish the presentation, because they were incredibly instrumental in the success of this study. The primary efficacy endpoint was essentially new HIV infections while people were taking the medications in a blinded way. And there was a safety endpoint that was also important that was grade 2 or higher clinical or laboratory events. And what that means is anything moderate or worse. Grade 1 is what's called mild, grade 2 is moderate, grade 3 is severe, and grade 4 is life-threatening. So we only looked at things that were moderate or worse for this analysis, for this study. Okay, these are the 43 sites that participated in the study. You can see in the US, there were 27 sites that participated, and you can see where they are with the little red pins that are in the map. We had sites in 3 countries in South America. We had 4 sites in Brazil, 2 in Argentina, and 5 in Peru. Some of those are overlapping in terms of the pins being dropped. That's why it only looks like there are fewer there, just so in case people were wondering. We had 1 site in Cape Town in South Africa, and then we had 1 site in Vietnam and 3 sites in Thailand. And those were the 43 sites that participated in the study. And these are all sites that have been funded by the NIH to conduct HIV treatment and prevention research. And that's specifically why these sites were chosen. And in fact, this was a subset of sites that the NIH funds where there was interest in population in conducting the study. So this is what the study design looked like overall. It's a little bit of a complicated slide, so I'm just going to walk people through it for a second. People who were eligible enrolled in the study, and then they got randomized, which means like flipping a coin, to either the Cabotegravir arm, which is in orange, or the Truvada arm, which is in blue. So let's take, for example, the Cabotegravir arm. That's the top half of the slide. The study has 3 phases. The first thing that happens is every day for 5 weeks, people took 2 tablets. One is the short-acting Cabotegravir tablet. That's that little thing that you see in orange that says Cab in it. And if you look below that, you'll see there's a Truvada tablet that's kind of gray, and that goes through step 1 also. And again, the one that is real is in color. The one that was a placebo is in gray. So for the first 5 weeks, people took a real Cabotegravir pill and a placebo for the Truvada pill. So 2 tablets taken every day. If people tolerated the Cabotegravir, the short-acting version, okay, then they moved on to step 2. And in step 2, they continued that placebo Truvada pill, but now they were given an injection, and the long-acting injectable version of the Cabotegravir. And that was every 8 weeks after the first 2 injections just being 1 month apart. And that whole portion, the oral and the injectable portions together, were set to last for 3 years. Before I get to step 3, let's just jump down to the bottom half of the slide in the Truvada arm. Basically, everything was the same except the Truvada pills were real, and the Cabotegravir, both the pill and the injection, were fake. So it was placebo pills, and the injection was a feeding solution that looked and smelled and for all the world felt like the active Cabotegravir, but it did not have any active Cabotegravir medication in it, and that was given at the same schedule. So the idea is you didn't know which medication was real and which one was the placebo. And then if someone, for whatever reason, decided they didn't want any more Cabotegravir injections, no matter which arm they were in, they got a year of open label regular Truvada for PrEP daily after they stopped their last injection. And the idea was because the long-acting injection took a while to wash out of people's systems, and you certainly don't want people at risk for HIV as that medicine is washing out of the system. So that's that step 3 that's over on the right side of the slide, and that was for both arms, not just one or the other. So I hope that study design made sense. It was complicated, and it actually was a really important thing to make sure that people entering the study understood what they were signing up for when they started, so no one was surprised or disappointed or confused as they started the study. So I told you that we had planned to enroll 5,000 people. The study stopped when we had enrolled 4,570. 4 of them were incorrectly enrolled, so they actually weren't eligible for the study. So that's why you see in this column that says total 4,566. You can see how they're broken out as to how many people ended up in each arm. Again, the orange is the Cabotegravir people, and the blue is the Truvada people. And what I want people to notice here that's, I think, is important is we set out to enroll at least 10% of the population transgender women, and we were able to enroll 12.4% of the population. So that's 567 transgender women. So we were extremely excited about that. We wish that there had been more representation of transgender women, but we did meet the metric that we had pre-specified. And we had said that at least 50% of the population will be young, under the age of 30. And we actually were 2/3 under the age of 30. So it was an extremely young population. And so that was really great. And you can see below that, the distribution of the rest of the ages, the regions that people came from. Again, remember, there was just one site in Africa. So that's why the representation from Africa is smaller. And then people's education levels and relationship status when they started the study. And that's just self-report. That wasn't confirmed anyway. That's just what people told us when we asked. So then in terms of race ethnicity, remember that we had pre-specified that we would enroll 50% of the US population who self-identifying as Black and African-American or African-American, and we just made it. So we enrolled 844 participants who self-identified as Black or African-American. And then you can see the race and ethnicity breakdown from the other regions. And in the United States, almost 18% of our enrollees were Latinx. In Latin America, you can see that 92% of people self-identified as Latinx from the sites in Latin America. So just a little bit more about the different regions. In the US, there were 1,698 participants enrolled. The average age was about 27. 61% were under 30. 7.4% from the US were transgender women. And I told you about our Black or African-American enrollment in the US already. In Latin America, and this is combined Peru, Brazil, and Argentina, the median age was 26. About 2/3 were under the age of 30, and 10.3% of their enrollees were trans women. In South Africa, so our one site, although they only enrolled 152 people, median age was even younger. It was 23. 75% of their participants were super young, under the age of 30, and 10% were trans women. We were really, really excited about that because cisgender men and transgender women who have sex with men in South Africa are populations who are really underrepresented in clinical trials and poorly understood from a research standpoint, and we need more enrollment from MSM and trans women in sub-Saharan Africa. Understand how to keep those people HIV uninfected in ways that are congruent with their lifestyles in the African continent. In Asia, median age was 24. 80% were under the age of 30. And look at this, almost 30% of the enrollment were transgender women in our Asian sites. So the majority of our transgender enrollment came from our Asian sites and our Latin American sites, but we did have almost 8% from the US. So that gives you a little bit of idea of who was in the study. And these are the results of the study. And this is a little bit complicated. So if you'll give me just a second, I'm going to walk through this. So overall, that's in the whole study, we followed people for 6,389 person years. And that means not everybody was followed for an even number of years. So if you look at the number of years that people were on the study medications and you add them all together, it's as if there was essentially one person followed for 6,000 years or 6,389 people followed for 1 year. It's sort of that kind of idea. And there were 52 incident HIV infections. That means 52 people who we're 100% sure did not have HIV when they came into the study. I'll tell you more about that in a second. Developed or acquired new HIV infection despite being in the study. The average length of time that any one person was in the study was about 1.5 years. And that gives you an incidence rate of 0.81%. Now, I know some people on the call may not have a context for, is that high, is that low? We were trying to enroll, or we estimated that the population that we would enroll would have an HIV incidence, if they weren't on any PrEP at all, of about 4.5%. Okay. If there had been a placebo arm in this study, which there was not, where people were getting both drugs, placebo, again, that didn't happen, we would know what the background incidence was in this population. But because we didn't do that, we don't know for sure. But to give you an idea of the ballpark of where people were starting from, we think it was about 4.5%. So the overall pooled incidence was 0.81%. If you look in the lower left corner of this slide, you can see how that broke out by the study arms. And again, orange is Cabotegravir and blue is the Truvada. So in the Cabotegravir arm, there were 13 new HIV infections. So the incidence rate in that arm was 0.41%. Again, imagine that we could be starting from about 4.5%. In the Truvada arm, we had an incidence rate of 1.22% because there were 39 new infections, okay? Again, starting from 4.5, it went down, we think, went down to about 1.22%. So the primary way we analyze this, because the only way that we can know for sure what happened in our study was from the data that we got from our study, right, is you compare the incidence rate of Truvada to Cabotegravir. Okay, now I'm going to ask you to look over to the right side of the screen. And what this gives you is if you divide that 0.41 by the 1.22, you get something called a hazard ratio. It's basically the ratio of how well one worked compared to the other. And what you get is 0.34. What that means is that there were 66% fewer new HIV infections in the participants who were randomized to get the Cabotegravir compared to the people who were randomized to get the Truvada. What you see on either side of that 0.34 are what we call the 95% confidence intervals. And what that means is there's a 95% chance that the true answer to this question of how does Cab compare to Truvada lies between those two goalposts, okay? Now, when we first presented these data in May, some of you have heard, well, it was non-inferior, but we couldn't say that it was superior. And what does superior even mean? So I've sort of labeled what that means in this picture to try and explain that a little bit. So non-inferiority is a statistical term that means not that much worse than, okay? We had decided before we started the study that if that right goalpost, the one that you see sitting at 0.62 right now, as long as that was to the left of 1.23, then we would be able to declare non-inferiority. Now, what that means is the result could be up to 23% higher HIV incidence in the CAB than the Truvada and still be called non-inferior. We didn't have to do that, of course, because you can see that 0.62 is way to the left of that non-inferiority line. The definition of superiority is if you exclude 1 from that goalpost, because 1 is the same, right? When you divide 3 over 3, that's 1. You divide 5 over 5, that's 1. So if the incidence rates were the same, then it should be 1. And so if your 95% confidence interval, that right goalpost, is to the left of 1, you're superior, okay? The reason there's also a 0.75 there is we went into the study sort of placing our bets, almost like you go to the horse race track and you say, what do you bet is going to happen? We bet that the Cabotegravir was going to be 25% better than the Truvada. That's what we thought we were going to see. And as you can see, that 0.62 is actually to the left of that. So it's not only to the left of non-inferior, it's not only the left of superior, it's the left of an even more rigorous boundary that we set before we did the study. So the statistical conclusion is it's superior. But what's really impressive about that is let's take a step back and remember Truvada. Truvada works really, really well if you take it as prescribed to prevent HIV infection. If you take it as prescribed for cisgender men and transgender women, it's more than 99% protective. So the fact that Cabotegravir was superior statistically to Truvada does not mean that Truvada is a bad drug. Truvada is a great drug if you can take it as prescribed. What this means is we have found something that works even better for people who have trouble taking Truvada as prescribed, or maybe they don't want to take a pill. So this is really powerful information, and it's super exciting that we have a really powerful new tool. But what I don't want people to take away from this is this means that Truvada is bad. Truvada is not bad. Truvada is a very safe, highly effective product. We just found something that works maybe for some people who aren't so good at taking pills, and that's super exciting. Okay, let me tell you a little bit about the Cabotegravir infections. And the reason this is important is overwhelmingly, the reason people get HIV infection when they're taking Truvada is they weren't really taking Truvada. So people don't take it as prescribed. It's very forgiving even so. But if you don't take it at all, or you take it very infrequently, then you basically don't have protection. We don't know a lot about the Cabotegravir and what's true or not true about it allowing people to get HIV infection. We know that there were only 13 new infections in this study, but what do we know about them? And so what I'm showing you here under the blue writing is, notice if you add up 2, 3, 5, and 5 there, that's not 13, that's 15. And that's because 2 of them, those 15, were actually infected before they ever got any study medication. We didn't know that at the time. We went back later and did super sensitive testing on their specimens from the day they came into the study. And so 2 of those 15 are not counted in the 13 that I'm telling you about. Of those 13, 3 occurred while people were taking the pill-based short-acting Cabotegravir. To me, that could mean that we need to know a lot more about how fast protection from Cabotegravir comes on board when you start taking it. We know with Truvada that it can take between 5 and 7 days of dosing before you get full protection, and that taking a double dose of the Truvada can accelerate your time to having protection on board. We don't know a lot about how fast you get protection from Cabotegravir as a pill yet. So that's something that we need to look into. 5 of the infections happened during the injectable phase. We need to know more about that, about what people's drug levels of the Cabotegravir were at the time they got infected. We don't know that today. I look forward to sharing that with you as soon as we know it. And then 5 of those 13 infections happened when people basically hadn't been on any Cabotegravir for like a year or more. So you can't expect that the drug is going to be protective if you haven't taken it in over a year. And we suspect that they're going to be very low, if any, levels of Cabotegravir left in those participants when we get that information. So of those 13, I think it's really, really critical that we get the drug level data and understand in a much more careful way. At least 8 of them, certainly all 13, are important to look at in detail. And in the Truvada arm, there were 42 infections overall. 3 of them happened before anybody took drugs, so I didn't tell you about them. 39 happened while people were taking the Truvada. And again, looking at the drug levels, both in dried blood spots and in plasma, is going to tell us a lot if we're right that these people actually just weren't taking their Truvada, because that's certainly the suspicion about why HIV was able to be acquired anyway. In case you're wondering if that 66% fewer HIV infections was generalizable across different populations, I'm going to ask you to look over at the little plot on the right side of this screen, because what this tells you, broken down by age, over 30, under 30, whether it was our trans women or our cisgender men, whether in the US it was people who had self-identified as Black or African American, or people who did not identify as Black or African American, and by region, all of the results were basically trending in the same direction. The study wasn't powered to give exact numbers for these different populations, but you can see it all favored the Cabotegravir and was ballpark of the same magnitude. We can't make statistical conclusions like this because it wasn't powered, but you can see that this was true across all the different groups that we had pre-specified. A lot of people say to me, well, how do you know how well people took the Truvada? And the answer is, I know this is a little bit of a complicated slide. I'm going to ask you to look at the multicolor graph all the way over on the left, to the left side of that vertical black line. So in 400 random participants who happen to be randomized to the Truvada arm, we have results already from about 372 of those 400 people. And the green, the light green and the dark green, tells you what proportion of people had adherence to the Truvada that should have been highly protective. And you can see at the top, the banner tells you that just about 3/4, 76.1% of those people should have been highly protective against HIV infection. You can see down at the bottom that there were 5.3% of people who didn't have any detectable drug in there. And there were another 7.8% who were taking it very infrequently. And then to the right of that vertical black line, you can see how that changed over time while on the study. And a lot of studies have seen those green bars get really small as people go to the right, so they sort of fatigue from taking pills. It seems like that happened a little bit if you look at the numbers across the top, but it didn't happen a ton. So that's interesting, and it makes you wonder if the support of being in the study actually was helpful for people in being able to take the Truvada and stick with it. So we thought that was really interesting. Again, the pictures aren't quite as pretty, but this is the equivalent of those green numbers that were on top. The proportion of people in each of the different pre-specified areas who should have been highly protected against HIV infection on the Truvada arm. So you can see that first set of red bars to the left is by region, and then we have the US participants who self-identified as Black or African American having lower drug levels on average than the US participants who didn't identify as Black or African American. You can see the Latin America participants having some lower levels. Didn't really differ so much by age, less than 30 or over 30. And our trans women participants, lower drug levels compared to the cis men participants. So in terms of safety and side effects, I'm going to ask you to look all the way over on the left, which to the left of that dotted black line, these are injection site reactions. So that's basically pain or swelling or tenderness at the site where people got the butt injections. And the colors are how severe it was. It's like a traffic light. So green is mild, yellow is moderate, red is severe. And you can see overall, the solid bar, that's the Cabotegravir people, almost 81% of people had a something after they got their injections. And very few of them were severe. Most of them were mild or moderate. And the hatched bar to the right of that one, where it's 31.2%, that's people who are getting the fake injection, the placebo injection. So you can see that just getting an injection, a third of people had some pain or discomfort, but none of it was severe. And then to the right of that dotted line, you can see that over time, it seemed like the injection site reaction reports went down. So that's good news. It seems like people sort of got accustomed to it. But what I will tell you is that 2.2% of the people who were in the active Cabotegravir said, this is so painful, I don't want to do this anymore. So there are some people that it's just too painful and they don't want it. And how bad that injection site reaction was really did correlate tightly with who stopped taking the injections. So people also have said to me, so I'm curious, I'm wondering maybe the low incidence rates for HIV that you saw in the study is because you actually were in really low risk people, even though you thought that you had really high risk people. And what I'm going to show you here is this is incidence of new other sexually transmitted infections, and these are really high numbers. They're not quite as high as they had in the Discover trial, except for syphilis, which is higher. But the rectal gonorrhea and the rectal chlamydia certainly indicate that there was a lot of folks having sex that did not involve a condom. So I don't think the explanation is because people were using a lot of condoms and were not certainly at risk for bacterial sexually transmitted infections. Of course, those things don't always correlate with HIV risk, but we think that they certainly are a marker for condomless sex. So I think this is an indication that this was a population that was having condomless sex. I think that's really important. This is just one really interesting piece of information. I know we're talking a lot about Descovy and Truvada, and which is better, and is there advantages, and kidney and bone, and weight gain. And weight gain is something that's really important, particularly for people living with HIV who are seeing this associated with integrase inhibitors, of which Cabotegravir is one. And what we saw in this study was a difference in the amount of weight gain in the two arms. So the Cabotegravir people gained about 1.3 kilograms per year, and the Truvada people gained only 0.3 kilograms per year. What's really interesting about that is if you look at the curves, in the first year, there's a huge difference. In the first year, the people on the Cabotegravir gained about 1.5 kilograms, and the people on the Truvada lost about 0.5 kilogram. But then after that, after the first year, basically they go in parallel. Everybody gained about 1 kilogram per year after the first year. So what does that mean? To me, it means that in the first year on Truvada, people lose a little weight and then they start gaining it again. And on Cabotegravir, they don't ever lose weight. They just gain at a rate that actually is comparable with a placebo from our phase 2 study. If you look over on the right side of the slide, you can see that in that study, which was HIV uninfected people getting Cabotegravir or a saline placebo, everybody, placebo or Cabotegravir, gained about 1.5 kilograms per year. So interesting. And this is just going to add another decision point in whether people want the long-acting injectable or they want to sort of get through that first year and see how it goes. So what are we doing now? We're measuring the drug concentrations to better understand why the people who got HIV infection in the study got it. In the Truvada arm, was it because they weren't taking it? That's our supposition. In the Cabotegravir arm, are they metabolizing it faster? Were they not taking it? In the oral phase, what was going on with the injections? We don't know the answer yet. And what does it mean for resistance? If people get HIV infection despite taking cabotegravir, do they get cabotegravir resistant virus? And what does that do to the ability to use other integrase inhibitors as part of HIV treatment down the line? And that's really important to understand. We just don't know the answers to that yet because COVID has slowed down all of our ability to get the samples. These are our conclusions, and I promise I will stop. Both drugs were highly effective for HIV prevention. The one that had the cab in it was superior to the Truvada arm. 66% fewer people got HIV infection if they were randomized to the cabotegravir compared to the Truvada. Despite the pain and the injection site reactions, it was generally well tolerated. And we really need to understand why the people who got HIV anyway got HIV and what we can do to prevent that, because even those 5 infections that happened when people were getting regular injections, we need to understand better. And what does it mean for resistance and subsequent treatment and viral suppression? This is the first long-acting injectable to show prevention efficacy in cisgender men and transgender women who have sex with men. So that's super exciting. But of course, to be fully excited by these results, we really need to understand if this works in cisgender women. And I hope everyone on this call knows this, but there is a sister study to HPTN 083 called HPTN 084 that started about a year after HPTN 083, and it's ongoing. And so we will have these data, and we're hoping that we will have an answer as early as November. But the data are the data, and we can't make something happen faster than it's actually happening. But I can assure you that people are looking at those data carefully as it goes on, and everyone's very excited for those results. I'm going to stop there. Really important, I told you at the beginning, it took a large number of people to do this study. It didn't just take a village, it took a metropolis. The NIH sponsored this. ViiV provided additional funding. The HIV Prevention Trials Network led the study, pharmaceutical support from ViiV and Gilead. Our partners at NIH, the HPTN leadership, the Operations Center, the Lab Center, the Statistics Center. We had an amazing study team. We had amazing investigators at all of our sites, teams at the pharmaceutical companies. And then here's what I told you, all the incredible people at all of our sites who worked like you would not believe to implement an incredibly complicated and difficult study. And of course, we could not do this, and we are eternally and forever grateful to the participants who took a chance on believing that we would do a study that would represent people who looked like them and get them new prevention options. And they believed in an experiment that they were a part of that now gave us this incredible result. I will stop there, and thank you so much for your
some information that I'd like to go through. So, one of the things that you've heard from Dr. Landowitz is he really went through all of the particulars about the study and and all of those other sorts of things. What I'm going to share with you guys a little bit more about today is about some of the community engagement work that was done with regard to this study, HPTN 083, and in particular, some of which was done through a body called the HPTN Black Caucus. Okay, how do I? So we'll start just by talking about the overall mission. And this mission is about the mission of the HPTN, the HIV Prevention Trials Network, which is all about discovering and developing new strategies to reduce the acquisition and transmission of HIV. The scope of work of the network is fairly broad. There are people who are doing clinical trials and research here all across the United States. And as you saw in the slide set that Dr. Landowitz showed, literally all across the world. So when we talk about some of the different trials that are being done through this particular network, there are literally things happening all, in every part of the world that you can think of at almost any given time. So, but one of the things that I wanted to talk a little bit about, right? So a large and, multifaceted study such as HPTN 083, really does come together and is only really successful when researchers and community people come together. And what happens in that process is we want to ensure that members of the community are really looking at making sure that whatever clinical trials or research is being done benefits as many people as possible without doing any harm. And so what you'll see here are 3 principles that are really, really important that we make sure that are included in the conduct of clinical trials, including self-determination, which basically means that people get to decide for themselves whether or not they're going to participate in a trial. And in order to do that, they need to really understand what the trial is going to involve and include, self-reliance, and then also principles about self-esteem. And so with regard to community programs, what you'll see here is that we wanted to make sure that people understood that that engagement of community is really, really important, right? So that we make sure that a particular study is that the research is desired. We want to make sure that it's not just sometimes you may hear the term helicopter research, where somebody comes from outside of a community because they have a particular idea that they want to do and study a particular population that might not necessarily align with the goals and objectives of people within that community. So we want to make sure that the research is desired. We also think it's really important that people start studies. So when we talk about recruitment and community engagement, but also retention, that they finish studies and that we support that. And then, of course, when it comes to understanding the data that's collected, we want to ensure that the data or results are relevant for as many people in the community or target communities as available. So with regard to the HPTN, what you'll see here are, these are the various ways that the network has or utilizes to engage with people in the community. We have community representatives who are participating as members of protocols. We have scientific working groups, which are opportunities where community people can actually come and participate in discussing and making, helping to influence decisions about the types of studies that are being developed, including a study like HPTN 083. We have a community working group, which is a very large body that includes community people in this particular study from all of the sites where the HPTN 083 study was conducted. Every single site had at least 2 representatives involved in the community working group. Of course, there's the HPTN Black Caucus, which I'll talk a little bit more about. Then, of course, you'll see here also that's really important are the local community advisory boards and stakeholders. And honestly, the most important are study participants. So when it comes to community, one of the reasons why community is really important is because of histories of medical mistrust with regard to clinical trials. Now, because within the HPTN Black Caucus, we really talk a lot about clinical trials and the engagement or inclusion of Black or African American people. I highlighted these particular 2 examples of things because these are some of the studies or experiences that some of us may not actually have been around for, but these are things that still influence the work that we all do today. One of them including the Tuskegee syphilis study. Now, what you'll see here is that it started in 1932 and the study actually continued until 1972. Yes, it was originally to look at the natural history of syphilis, but the way that this particular study communicated information to people in the trial was not exactly what people were understanding. And so once there was an appropriate treatment that was available, then rather than say to people in the study, oh, well, we now have penicillin and we can treat it, they continued not to treat it. And so these are examples of, when we're talking about clinical trials, that sometimes people in the community say, well, I don't want to do this because of this or because of that. If you do any work with you know, some of the biomedical stuff, or you may have seen either the book about Henrietta Lacks or the film about her as well. She was a woman who had ovarian cancer or uterine cancer, I believe. So they took some of her cells, and these cells continue to grow and multiply. And even though she's no longer with us, we still actually have this line of cells, and we still use them in clinical trials to this day. You know, and unfortunately, there was no consent that was obtained when, you know, at the time in order to get the cells or to continue to use these cells. So these are some of the reasons why organizations or bodies such as the HPTN Black Caucus are really, really important, because looking at these types of examples, and we recognize that not just Black communities, but a lot of racial and ethnic minority communities have been underrepresented in trials, both biomedical and social science research, but you know, in particular HIV clinical trials, and there are a lot of reasons behind that. But we also understand that if we don't have people who are willing to engage in clinical trials, that when we do have some outcomes, we don't know for sure, we may not necessarily know for sure the degree to which the outcomes are going to be as appropriate or effective for our communities as well. So that's kind of where some of the work of the HPTN Black Caucus comes into play as partners within research. And, you know, and one of the things that we were able to do as partners with the 083 study team was really work with Dr. Landovitz and his colleagues, and you know, in a reciprocal way that really supported engagement of people to be able to participate in the trial, and to feel, hopefully, that the trial that they were participating in was appropriately designed and met the needs of the people in the community. Very quickly, I'll talk about who we are as the HPTN Black Caucus. So the HPTN Black Caucus has existed within the network since 2008 and was originally organized around a particular study that was looking at some interventions regarding HIV that were oriented around Black same-gender loving men in a variety of places across the United States, including here in Los Angeles. The body has continued and has grown to embrace multiple clinical trials that are being done throughout the network, and to provide you know, support for integrating you know, community trials and the voice of community, so that they're both ethically competent, but also scientifically sound as well. And we communicate both out to the community and also from the community back to the study teams. As far as leadership goes, I just wanted to touch on this very briefly. You know, the original chair was Dr. Sheldon Fields, who's still involved with the network. Many of you may know Christopher Chauncey Watson, who was chair for a while as well. I've been the chair right now for a few years, and we also have a co-chair, which is my colleague, Craig Hutchinson. You'll see here a number of trials that the caucus has been involved with, including HPTN 061, 073, which was a PrEP study, the AMP study, or 085, 078, which was a study looking at linkage to care and engagement in care for people living with HIV, sort of a case management type of approach. The study that we're talking about this afternoon, 083, and a study that's currently in development that's looking at getting to zero or eradicating HIV in populations of Black men in the South. And that study is currently being developed right now under the leadership of Dr. LaRon Nelson. So with regard to 083, and I know that we have, I'm trying to be mindful of the time, but so, you know, Dr. Landovitz mentioned some of the recommendations or targets that we had, and it is true, all of the reasons that he articulated about why it was important that we have these targets of 50% engagement of Black or African-American men in the United States in a trial such as this, and people under the age of 30 and transgender women. A lot of these recommendations, particularly the representation of African-Americans, came out of some work that the caucus had done as a part of the HPTN 061 study, and our strong recommendations that we continue to make for studies moving forward domestically here in the United States through the HPTN. We did some cultural competency training. So it was really, really important because there were so many people who were coming into the network for a study such as this, some of whom had done work historically with African-American populations, but some who may not have. So it was really important to lay a foundation of some baseline cultural competency training. And that was done through a module called The Presence at the Table that really explores things, not just culture, but it also explored things such as implicit bias, institutional racism, and power dynamics, including race, gender presentation, and all of those other sorts of things, and recognizing who has power and what that means for the way that we work or how we engage with people. There were ongoing consultations with folks in the study team, and then also technical assistance and support for recruitment and retention. And some of that was also done with the community working group. And also I wanted to acknowledge the community programs team. I think this is my last slide. I just wanted to say very quickly that when we start talking about research that we're doing with, in this case, Black communities, but also I think all racial and ethnic minority communities who are at risk or unequal risk of HIV, the conduct of clinical trials should be timely. It needs to be done when it needs to be done. And while I think that it doesn't only need to involve racial and ethnic minority researchers, I think that it's also really important to make sure that if, for example, if you're looking at Latinx populations and communities, that you have Latinx population representatives in the research and design team. So leading some of the efforts, and same for for African-American populations, same for, gender non-conforming populations as well. If you have a study that's focusing on a population, that you need to make sure that there are researchers and stakeholders who are engaged at all levels of the project. We need to understand and ensure that there's a diversity so that, when we looked at all of the places where this study occurred, Black people, say for example, a young Black gay man in Los Angeles may have a very different lived experience than his counterpart or colleague in Atlanta or someplace else in Jackson, Mississippi. And it's really important that studies like this pay attention to all of that. We need to make sure that there's sufficient funding so that we can do this work and do it well. We need to make sure that there's partnership with other organizations and that we use culturally appropriate or relevant frameworks or models or language as well. And that also we really need to be innovative. And one of the things that I will say that the partnership of the HPTN Black Caucus with the 083 study team, working with Dr. Landovitz was a great opportunity, and it was very reciprocal and and allowed for a lot of creativity to happen in some of the work that became the 083 study. Final thoughts. These are my final thoughts, essentially, that it's really important to make sure that community is included. This particular trial is a great example of that, but within the HPTN, with all of the studies that we do or that we're trying to conduct, it's we're very, very mindful of community engagement and keep making sure that people and the voice of community are included. And there are ways that you can get involved. You can be a study participant. You can get involved as a community member, as a representative within your community, as a member of an advisory board, or other aspects within the network. I think with that.
Thank you, Chris. My name is Kamal. I'm originally from Washington, D.C. Graduate of UMBC in Baltimore County. I moved out here a year ago, just over a year, last March. So I'm over just about a year. Professional dancer. I moved out here for those exact reasons, to start a career, but not only that, a better career that I had in DC. I was a part of this study. It started for me in 2017, like late November 2017. That's when the ball started rolling for me. So I started off at the George Washington University site. Really good site. The people were great there. They took care of me. They always were there if I needed anything. And then I transferred out here last March, and it's been great. great group of people. They feel like family between Chris and Susan and Skylar. Like, it's definitely been a great transition. It was very easy. I didn't think they were going to be able to transfer me because, you know, I wasn't aware. I was aware of all the sites, but didn't know the chances of me moving to California. They would say, oh, we have one in California. So that worked for me. What made me decide to join this study was just, you know, I know that medicine and the medical field is always being progressive. And the fact that there was an opportunity to possibly not have to take Truvada every day, or me being a part of a study helping with the results, that's what drew me to it. I wasn't taking Truvada before this study, so I was aware of it, but all it was very new to me. But again, I just felt like it was very important to just contribute myself to it, and me being in it can help with, you know, research or anything. I just thought that was a great thing. But again, transferring, it was very easy. It wasn't as much of a hassle. It was the same things, like my visits consisted of the same. It was always a great environment. I always felt like I was an individual and I was treated like family. Anytime I had an issue, or if I felt like, you know, worried about something or anything, they were always available to me. There was a hotline I can call if it was something. And usually anytime I would email them or text them, I would get like an instant response, which is something that I really appreciated. But I would say the most memorable experience for me is honestly just the people and being able to work with the workers. I was surrounded with great people at George Washington. You know, they were always flexible with me when I couldn't get to an appointment or something. They were always active, always asking me about like my career choices and all that, like how's everything going? And even transferring out here just to come to a place that I didn't start with, but feel like, you know, I just got in instantly and just everything ran smooth. Like they felt like a family. They're always asking about me, checking on me, asking, you know, how's dance, how's this? So it's been very, very great. It was just a great experience for me. I don't know if there was any questions for me, but that's pretty much like everything for me. Yeah.